Good day, my dear postgraduate students. Tell me frankly, how comfortable are we with the technical topics related to physics or chemistry? Personally, I am not so. That is why I took to biology and then to medicine, landed here. But today, this is all haunting us even in the examination. The topic for the day is electrophoresis. One of my postgraduate students had requested me, and I took a plunge into it. The introduction. Tiselius was a biochemist in Sweden who first designed it in 1937. In electrophoresis, various biomolecules such as proteins, peptides, nucleic acids, and nucleotides all get separated. In an electric field, they migrate towards either the anode or the cathode depending on the charge that they possess. Molecules with a similar mass and charge move together. The quote again. Nice one. Separation is the absence of love, and love is the absence of separation. I think this author must have very well felt it. Definition. A charged particle placed in an electric field migrates towards the anode or cathode depending on the net charge it carries. In other words, when an electric field is applied to a solution of ions, it makes the ions move. Basically, it is a process of separating the constituents of a solution which are charged. So whatever be it, you people will have to explain it. It is not very difficult. This is just an example, the capillary electrophoresis. So there is a negative charge and then a positive charge. There are the ions which are placed over here and then they move towards one pole. Tiselius, he initially used a liquid medium which was not very successful, but subsequently solid support media with buffers had produced better results. What are the factors that support the electrophoretic mobility? It can be the size, the shape of the molecule, the strength of the electric field. 3S probably. This is again a very important table, very easy for us to by heart. These are the various components in an electrophoretic apparatus. Buffer, it carries the current and maintains the pH. Wicks contains supporting medium for completion of the circuit. Support medium, it can be a matrix in which there is separation of the particles. Cover, to reduce the evaporation. Power supply, it is the energy that is required for the charged particles. And densitometer is quantification. So very simple, and I would like you people to kindly by heart and write this. Not very difficult at all. Whatever you can recollect, you can write. We are quite familiar with this hemoglobin electrophoresis. Look at this one. So there is a negative charge and a positive charge, and you find that the origin of it, the sample is placed over here, and the various hemoglobins are moving at different speeds so that they get separated. So in this one, A2CU and STG, F and A. I used to tell our friends, look at this mnemonic, A2, which means V2. Get into a train and say, see you to our friends. And the train starts moving. It has got a rhythmic movement. It says STG, 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 and it keeps moving. It goes to the fag end of the journey, FAG. We get down at a station called R.A.T.S and we go home. Hemoglobin H. So that is a mnemonic that I deciphered for the cellulose acetate electrophoresis. So here, the various hemoglobins, hemoglobin F, for example, this can be in the case of a neonate, and In a case of sickle cell disease, there is only hemoglobin S. In a trait, there can be the sickle cell hemoglobin S as well as the hemoglobin A, which protects. So that is how it is being separated. So some of the diseases, I'll go to the other diseases in the forthcoming tabular columns. DNA electrophoresis. In this one, there are two components. One is the fluorescent under the UV light, and second one is a stain component. So it is the visualization of the proteins in a paper or gel. Here I think it's a gel. So the gels are soaked with ethidium bromide and it is subject to the ultraviolet light. I find that there is a fluorescence and then I can see the separation. Otherwise, it can be stained with fumacy brilliant blue, as a result of which I can see the blue color components which are getting separated. So it can be a nitrocellulose, or sometimes for antibodies we can use the Western blot technique. A fundamental one, this you can practice explaining by yourself. Negative charge, positive charge. And there is a gel. And there is a solution over here, a buffer that is meant for the suspension of all. This is the distance between the two. And after some time, I find that there is a gradual separation. These are the wells in which the samples are being placed. Multiple samples can be placed simultaneously, and depending on the density and the component, they get separated at different levels. This is a paper electrophoresis. Again, this is a thin paper that has been kept over here, the filter paper, and the Wattmann's filter paper is set. And there is a positive and the negative one, and the buffer solutions over here. And there is a movement of the bands that can be found in this. This is one of the earlier methods of electrophoresis. This is a previous picture. Again, I'm representing the DNA sample. There is a cleavage and it is loaded over here. The DNA travels. There is a separation of the various components by means of the agar gel electrophoresis. A basic principle in a case of a capillary. So this is a capillary, a sample is admixed over here and it is traveling. A detector is used to find out where exactly it is. It is very rapid and can be performed for small tests. So classification of the types of electrophoresis. So this again, the negative and the positive, I would like you people to kindly describe it. A buffer solution is here and these are the wells and the entire thing is being covered. A power supply is required and there is a transport that is being made. Zone electrophoresis is the most common. It can be applied as a spot or a band on chemically inert homogeneous supporting medium that should not have any charge on its own. And you find that the components are divided into zones or bands. So this is fundamental, the most common type of electrophoresis. The types of electrophoresis kindly go through. Paper electrophoresis takes a very long time and there is a mixing between the zones. It has been replaced by other media. Cellulose acetate, there is a minimal absorption or sticking and a clear separation. Good. Agar gel, transparent material used for immunoelectrophoresis on microscopic slides. Starch grains, prepared from whole starch as such. It is used for larger molecules like isoenzymes. Then polyacrylamide gel as well as the native polyacrylamide gel. They are transparent, scanned, and seen as UV light or obvious material. Fine resolution complex mixture. So these are the various types of electrophoresis. You people can mention a few. And there are the other types also. Isoelectric focusing, immunoelectrophoresis, high voltage, pulse field. For a good separation, capillary I have mentioned already, 2-dimension. Usually in a separation, it will be in a single dimension along the length. But here, after some time, you find that there is another dimension also. That is why it is called as 2-dimension. So I am finding the material getting scattered as such, which can be read by the physicist rather than us. This is of paramount importance, the hemoglobin electrophoresis. Earlier, I was thinking only hemoglobin S was most important. See what is happening. If your own blood is given, normal blood, the different hemoglobin fractions can be identified. In a cord blood of the patient, there will be the hemoglobin A2 and the hemoglobin F. So this will be important. So hemoglobin SC disease. So there will be the hemoglobin C as well as one gene product, the hemoglobin S. So they will have to be separated for identification. Beta thalassemia, you find that there is a marked elevation of hemoglobin F and A2 in the absence of minimal hemoglobin A. Hemoglobin H, there is a decline in hemoglobin F and A2, and this is in a case of hemoglobin H disease. See the different types of hemoglobin electrophoresis, the components that are being separated. It is continuing. This you people know, valent for glutamic acid, hemoglobin S, sickle cell anemia. Beta thalassemia, F and A2 fractions are there. And alpha thalassemia, there will be the absence of A, F, and A2. And when everything is absent, there can be the presence of hemoglobin H or parts. And what is the specimen or the sample that is required? It can be done in the serum, plasma, whole blood, hemolysis. The blood is lysed. Nucleic acid, protein extracts, products of PCR, and sequencing experiments. This I think we will have to buy. The factors that are interfering can be the heat, absorptive factors. It makes the components get stuck to the medium. And electroendosmosis. Endosmosis is again a perpendicular permeation that should be avoided. Gel electrophoresis is mainly used for DNA sequences, northern and the southern blotting and the restriction mapping. Applications in protein study will be determination of the molecular weight and subunit stoichiometry. What are the basic applications of electrophoresis in medicine? Forensics for the DNA fingerprinting. Molecular biology, again to separate and organize DNA and RNA. Genetics for DNA cloning. Virology to diagnose various strains. Biochemistry for the cellular components, mainly proteins. Haematology, the hemoglobins. Pharmacology, the drugs such as steroids and antibiotics. Vaccines, DNA sequencing. Western blot for the diagnosis of HIV and other infections. And how do you identify actually? There can be a stain that can be used. Initially, I showed you people the Kumasi blue, but there are a lot of other stains which can be used. Easy to remember. Fat means Sudan black, oil red wool, we already know. Glycoprotein, PAs, we already know. Then the nucleic acid, we'll be using the silver stain. Others you people can read. Hemoglobin ferricide. So most of the things you already know. It is put in the form of a table. Kindly read it. This is a Western blot, gel electrophoresis. This is a bacterial sample that is over here. There is a lysate extracts, and this is a gel cassette kept in an electrode chamber, and this is placed in a tank. Look at this over here. There is a sample. Multiple wells are over here. The cathode buffer and the anode buffer is over here. And then there is a movement that is being depicted by means of the various colors of the fluorescence. So this we have already seen. So there is a terminating electrode and a leading electrolyte. Negative and the positive charges, and there is a movement, and there are boundaries to decide to what extent they have moved. The references are STAT-PEARLS electrophoresis. I would like you people to kindly go through. In fact, the STAT-PEARLS gives a wonderful information for many a topic that we people need for the examination. And ever has it been known that love knows not its own depth until the hour of separation. Khalil Gibran, a hero of mine. Thank you for your patient listening.